Hi, good evening, all of you. Hi, good evening, all of you. Hope you are able to get my voice. Hope I am audible to all of you. Please confirm. Confirm in the chat box. Hi, good evening, all of you. Please confirm in the chat box so that we can move ahead for the discussion. Okay, I think I am audible and voice is clear to all of you. So, good evening, all of you. So let us discuss about our today's topic, and today's topic is bracketing and matrixing design. Basically, bracketing and matrixing design, this is a concept in a stability study, and this concept is as per the exactly as per the guideline ICHQ1D. Yeah, thank you, Emmanuel, for confirming. Thank you so much. So today we will be discussing about bracketing and matrixing design. This is really very helpful for reducing the analytical load in the quality control laboratory. And one more thing, it is not only reducing the load, but we can use the same data to submit the regulatory agency, and also can be used for shelf life determination of the particular product. So that is the reason this guideline is very important. Many companies are really following this guideline and can and get benefit from this guideline. So let us discuss about this training about this topic. So what we are going to discuss today, so we will discuss about the brief about this concept and the strategy about the guideline and all. Then there will be short information about me, a couple of slides will be there. Then there is a disclaimer by the guideline itself. So guideline has provided some clarification about that how it should be used and how the example should be considered. So that is what the clarified by the guideline ICHQ1D. So that we will be discussing. After that, where we can apply this reduced design. See, basically, you may be aware about the ICH Q1AR2 for stability studies. You know, so the point here is when we are discussing about the reduced design for stability studies, so where we can apply this strategy or thought, that we will be discussing under this head. After that, we will discuss about the bracketing. So we will have a different discussions, special discussions on bracketing, and a special discussion on matrixing. So under bracketing, there will be some design factors like strength, container closure size or feel, that we will be discussing, followed by the practical example of bracketing, how we can implement that example and how we can put into the stability study protocol, that we are going to discuss. After that, we will discuss about the matrixing approach. What is the meaning of matrixing? Similar way to bracketing, what are the design factors for matrixing and what are the different reduction plan, means test reduction plan, that we will discuss here. So in matrix, there will be some test reduction plan. So with that, we will have some practical examples of a test reduction plan with the matrixing. Then we will have a applicability and degree of reduction under this matrixing stage, that where we can apply and at what extent we can reduce. That also we will discuss under this head. Followed by, as this is a reduced testing, so what are the potential risk with the matrixing approach? There are no much risk with respect to the bracketing approach. We can smoothly we can follow immediately from today onward. Not much justifications or risk assessment is required, but for matrixing there are some potential risks. So that we will be discussing what are the potential risks with the matrixing approach, followed by the question and answer sessions. So I just want to request all of you that if you have any questions, so please put your questions in the chat box so that I will reply to all your questions, okay, at the end of the session. Also, if anyone is interested in a training material, so please request me now only in the chat box, so that at the end of the session I will share the link for downloading the training presentation. After that, I will be not having a training presentation copy. So please, this is what my request to all of you. And now let us move to the our actual training. And as per the agenda, there is a short brief about me. So my name is Hitendra Kumar Shah. I am a CGMP compliance consultant, trainer and auditor from NADH plus GXP compliance services. I am having more than 23 years of experience in a pharma in different domains. Further, I was a quality head, QA QC head for many companies. I face a lot of audits like US FDA, UGMP and all. I have audited more than 500 sites till date and trained more than 5,000 candidates approximately, of course. This is just a brief about me. This is the major work experience. I started my career with a Sun Pharma, then I work with the Blue Cross Laboratories, then I work in Alembic Pharma. I work in a Zydus Cadila as a compliance head. I was in US Vitamins. I work in some multinationals like AstraZeneca, Sanofi, Mylan, and since more than five and a half year, I am an independent compliance consultant. You can say the CGXP compliance consultant from NADH plus GXP compliance services. So this is what about my short brief, and before going ahead, just I want to share the disclaimer that whatever the details I have taken, it is from the guideline, and if you want to implement this, please refer the current version of the guideline, because this training session will be in the YouTube for a long period. So there may be possibility that the guideline may update after some time. So please understand that whenever you want to implement, you please follow the current guideline requirements. Okay, and now let us start our today's discussion about the brief of uh, you know bracketing and matrixing ICHQ1D for the stability studies. So there will be some time required, the delay will be there. So when I will change the slide from here, it will take some time. Okay, so there is some peculiarity between the other regulatory guidelines and ICH guidelines. So I will just brief about this as a short introduction about our today's topic. So basically the other regulatory guidelines, what they do, they develop the guideline and then they share to the common people or the industries to and take their they share the draft guideline and they take the comments from the the experts or comments from the pharma professionals. But here the ICH guideline is developed by the EWG, expert working group. So who is the involved in the expert working group? Expert working group means the regulatory experts, means the regulatory bodies experts, the people who are working in the regulatory authorities, they plus industrial experts both come together to form a expert working group, and then they develop the guideline, and after that they share the draft guideline to all the people for the comment. So this is the novelty of this ICH guideline, that it is not only designed or drafted by the regulatory people, but regulatory authorities, but it is developed by the expert working group where they take the input from the industrial experts also. So under ICH guideline, there are 4 categories: quality, safety, efficacy, and fourth is a multidisciplinary. And we will be discussing about the quality guideline, correct? Hope you are getting my point. So basically when we discuss about the ICHQ means quality guideline, so there are total 14 guidelines: ICHQ1, Q2, Q3 to the ICHQ14. And ICHQ1 is specifically for the stability studies, like ICHQ1A-R2 is for stability study of drug and drug products. ICHQ1B is for photostability. Similar way, ICHQ1D is for the bracketing and matrixing approach for the stability studies. And today we are going to discuss this ICHQ1D of the ICH guideline. Hope you are with me. Again, I am requesting, if you have any questions, please put your questions in the chat box. I will reply to all your questions at the end of the session. If you are interested in the training material, training presentation copy, please put your comment in the chat box. I will share the link so that from the link you will able to download. Okay. So guideline, ICHQ1D itself has given the clarification or disclaimer to all the people. What is the disclaimer? That this guideline provides the bracketing and matrixing study design. So they have given some designs for bracketing and matrixing. specific principles are defined in the guideline for situations in which bracketing or matrixing can be applied. So where we can apply the bracketing or matrixing, that situations are shared or recommended by the guideline. But very important thing is, whatever the examples are given or design examples are given, these are for the illustrative purposes. So you should not directly copy and paste. It may not be suitable for all the cases, you know. You need to evaluate and then you can implement at your end. That's it. That is what the clarification provided by the ICHQ1D guideline. So I am just making you aware about this. So many people think that it is a displayed in the ICHQ1D, so directly we can implement, we can put in the stability study protocol and we can reduce the testing. No, that is what the clear cut given. So because you know there are a lot of parameters are there that we are going to discuss step by step during our today's discussion. So my point is you need to apply your thought process and the product nature, equipment, process, so many parameters we need to evaluate. So that is the reason the guideline is giving the clarification. Now, applicability of reduced design. So where it is applicable to reduce the testing or reduce the stability design, that is what is given here. First of all, very important, first sentence is very important. You can go through this. The use of any reduced design should be justified. Very clear. So whatever you are doing any reduced design, it should be justified. Now it is clarified below. Reduced design can be applied to the formal stability study of most types of drug product, although additional justification should be provided for certain complex drug delivery systems. So if the drug delivery systems may be, you know, drug device or it is drug related to the devices and all, you need to have some additional justification because you know the releases will be key parameters there. For the study of a drug substances, matrixing is of a limited utility and bracketing is generally not applicable. For the study of drug substances, matrixing is of limited utilities and bracketing is generally not applicable. So why it is generally not applicable, we will discuss when we will discuss about the bracketing and matrixing, then you will come to know better way. But this is what recommended in the guideline. Hope you are with me and you are getting the point, right? So this is what about the applicability of the reduced design. Now let us discuss about the bracketing. So now we will first focus on the bracketing and then followed by the matrixing. Okay, and in the bracketing when we will discuss the requirements and all, followed by there will be some example. Okay, many companies are following this, very simple bracketing. And whatever I am explaining you, it is in line with the guideline, current guideline requirement. Okay, bracketing is the design of a stability schedule such that samples on the extremes of certain design factors. Means extremes means what? Strength, lowest strength, maximum strength, or and size, container size means smallest size, biggest size. So this point we have to consider. The designs assume that stability of any intermediate level is represented by the stability of the extremes tested. That means what? If I am testing the smallest dose and testing the largest dose, then it will assume that the intermediate middle dose will be comply the things. That is what the expectation here, or a clarification is given under bracketing. So design assumes that stability of any intermediate level is represented by the stability of the extremes tested. So if stability of the lowest strength and highest strength or lowest pack and highest pack is complying, then automatically the stability study of an intermediate level will be complying. So that is what the thought process behind the stability study for bracketing approach basically. Hope you are with me, right? So let us discuss about the strength. So bracketing, when we are discussing, so strength and packaging, two major aspects are there, right? So bracketing can be applied to the studies with multiple strengths of identical or closely related formulations. So when you are discussing about the bracketing, so bracketing can be used where they have the similar, very identical, you can say the strengths and closely related the formulation. So formulation should be exactly same or minor changes can be justified. But again, it has to be justified, okay? For example is also given here. You can go through this. Capsules of different strengths made with different fill plug sizes from same powder blend. Means what? Powder blend is same, used to fill different strength of the capsule. Powder is same, the granules are same. With the granules are filled in the capsule. If the less granules are there, the dose will be small. More granules are filled in the larger capsule size shells, then dose will be high. That means what? Powder blend is same, only the fill plug in the capsules will be different. In that case, we can use the bracketing with respect to the strength factor. Or for compression, if the granules are same but we compress at different weight. So if we granules are same but we compress suppose at 100 milligram and we compress at a 200 milligram, so it will have a different strength. So in such a cases we can make a use of bracketing approach. Hope everything is clear, right? I am making it very simple language, easy to understand language, okay? In cases where different excipients are used among strengths, bracketing generally should not be applied. So here the basic is what? The formulation or the formula should be identical or closely related. But if the different excipients are used, so formula will be different, formulation will be different. So you should not generally follow the bracketing approach for such a type of products. But if formula is same and exactly formulation is similar, then definitely you can go ahead for the bracketing approach. Hope this is clear. So now the second design parameter for bracketing is the container closure sizes, right? Second design parameter for bracketing is the container closure sizes. So same thought is applicable for container closure sizes also. Bracketing can be applied to the studies of the same container closure system where either container size or fill varies while other remains constant. Simple wordings, right? If a bracketing design is considered where both container size and fill vary, it should not be assumed that the largest and smallest containers represent the extremes of the packaging configuration. So if the packaging components are different, you should not just assume that the largest and smallest containers will represent the extremes, because there are a lot of parameters when we are considering the container closures, right? It will there are different parameters like the free space or vapor permeabilities and all. So many parameters are there. So you should not consider that that if you have a different packaging components, so definitely it will comply the things. And that is what here explain the thing about the container closure in the next slide. Okay, please wait for 2 3 seconds so that the next slide will be appearing on the screen. If you have any questions, you put your questions in the chat box. At the end, I will reply to all your questions. So what bracketing with respect to container closure sizes, I was explaining you about this care, that is what displayed on the screen. Care should be taken to select the extremes, right? That is what our expectation when we are discussing about the bracketing. So extremes by comparing the various characteristics of the container closure system that may affect the product stability. So what does it mean? These characteristics includes container wall thickness, closure geometry, surface area to volume ratio, head space to volume ratio, water vapor permeation rate or oxygen permeation rate per dosage unit or unit fill volume, etc. So these points also you need to consider when you are considering the bracketing for container closure sizes as the design parameter for bracketing approach. So you need to consider the wall thickness of the container. So thickness wall thickness of the minimum and maximum size should be same. Closure geometry should be same. Surface area to volume ratio. So whatever surface area means the diagram is presenting the same is surface area to volume. So headspace should be similar. Headspace to volume ratio. Then water vapor permeation rate. So how much water vapor is permeated and all that rate. Oxygen permeation rate per dosage unit or unit fill volume. So these parameter you have to evaluate when you need to think about or plan to make a bracketing approach. Correct? Simple things. Now I will take some examples so that it will be very easy to make you understand. Very simple example I am putting here. So you will come to know in a better way. Okay. If you have any question, please put your questions. Okay. So that, I will respond to your all question. So a simple bracketing design approach is explained here. So what happened here? There are suppose three strengths. Okay, 50 milligram, 75 milligram, and 100 milligram strength is there, and each strength is having a three pack styles: 15 ml, 100 ml, and 500 ml. And for each pack style, there are suppose three batches, right? Means 50 milligram. Let us say example 50 milligram having three packs, each pack will have a separate batch. So like that is a scenario displayed here. So three different strength, three different packs, and each having a three different batches. So that is what displayed here. You can see now here point is what? Strength, so there is a exact same formulation, then only we will able to use this bracketing approach. Correct? So as there is exact formulation, so what we can do, 50, 75, 100. So 50 and 100 is the extreme, so we will focus on 50 and 100, and we will consider 75 milligram as an intermediate level. So it will represent the 50 and 100 milligram will represent the intermediate level. So if 50, 100 milligram is complying to the requirements of stability study, 75 will also follow the same, because it will represent the intermediate level. So that is the way. Intermediate level means intermediate strength or pack style. Intermediate stability study is a different subject. Okay. Intermediate conditions, storage conditions. I am not telling about the storage conditions. Okay. I am telling about the intermediate level of the strength. 50 milligram and 100 milligram. So intermediate strength is 75. So you can skip 75 milligram strength. That is the first point. Second point is now each strength is having a three pack style: 15, 100, and 500 ml. So when we are discussing about the three pack style, so here also you can skip the 100 ml and you can perform the stability study as per ICHQ1D for 15 ml and 500 ml. And that way, the simple plan is prepared and it is displayed here. T means test samples to be tested. So and blank means no need to test the sample. This is the way you can perform the bracketing approach. Let me tell you that this approach is widely used by many companies when you are manufacturing the product, and I am telling about the regulatory regulated market. Okay, so many companies are following this. If there are multiple strengths, many products will have the multiple strengths, like alprazolam, or you can say a lot of even the some are metformins, or so many other general products also. So whatever the products are there, if they have the multiple strength and they are stability you are performing, and when the stability study is going on, you are planning to have a stability studies for submission to the regulatory agencies, then this approach is widely accepted by the regulatory authorities when you are having this bracketing approach, and you can reduce the testing. Very, very clear. I am telling you, this is the widely followed approach. Okay, bracketing is widely followed approach. Matrixing is somewhat tricky. Bracketing is very simple and easy, widely accepted approach, but matrixing is somewhat tricky. So bracketing is very easy now. Up to here it is very clear, right? So now let us discuss about the next, that is a matrixing. So what is meant by matrixing? Let us discuss about the matrixing. So hope bracketing is clear to all of you. I presented one example. Similar way now we will discuss about the matrixing. If you have any questions, please put your questions in the chat box. I will reply you at the end of the session. If you need a training presentation copy, please ask me in the chat box. I will share a link in the chat box at the end. Okay, you can download the training presentation. After that, I will not be having the training presentation with me. So matrixing approach is what? It is a design of a stability schedule such that a selected subset of the total number of possible samples for all factor combinations would be tested at a specified time point. So here we are focusing on testing. There what happened? We directly skip specific pack. In the bracketing what happened? We directly can skip the particular pack, particular strength for even we will not perform any testing, we will not perform any stability study, right? Here what happened? We will reduce the testing itself. We will reduce the testing itself. So how? There will be some subsets of samples. So don't worry, I will have the example so that you will have the more understanding about matrixing. But there will be some subsets of sample. How? Like one sample, it will be tested at 0, 3, 6, 9, 12, 18, 24, 36. Let us say 36, 3 years study. So these are all the subsets of samples. 0 to 36, means initial to the end of the stability study. These are all the subsets of the sample. The design assumes that stability of each subset of samples tested represents stability of a samples at a given point. What does it mean? It means that each sample, suppose there are 3 batches, right? Each batch will have a different time points. So, for example, suppose the first batch I am testing at a 3-month. There may be possibility that you may skip the second batch at the 3-month. You can directly go the next, so that way. You got my point? So the point here is design assumes that stability of each subset of sample means what? Let us take example 3, 6, 9, 12. Okay, batch number 1, batch number 2, 3, 6, 9, 12, batch number 3, 3, 6, 9, 12, right? So batch number 1, if you are testing at 3 months, then it is considered that batch number 2 and 3 will behave the similar way how the batch number 1 behaving at the 3-month. So that way, that thought is used. So this is called the subset of samples, and this thought is used when we are discussing about the matrixing approach. Correct? Now here, some information is given. This is very, very important. Each storage condition should be treated separately under its own matrixing design. Means what? If you are not aware, you can go through the ICHQ1D AR2 for stability study. If you are not aware, I will share a link. You can go through the link. I will share the link in the description section. You can go through this. What are the different storage conditions? Okay. So there are accelerated conditions, there are intermediate conditions, there are long-term condition, right? So when you are performing the matrixing, you should not skip the particular condition. You cannot skip. But in each condition, you can follow the matrix approach. So each storage condition you have to respect and you should treat it separately. That is what the information is, very clear information. Matrixing should not be performed across test attributes. However, alternative matrixing designs for different test attributes can be applied if justified. What does it mean? It means that when you are performing the matrixing, already you are reducing the testing. Reducing the testing means what? Reducing the samples to be tested, right? But if under one sample there are 5 tests or 6 tests, related substances, identification, assay, dissolution, or maybe content, whatever, content means assay, potency, or other tests also. You should not reduce the test. You should not reduce the test under that particular sample if you are performing the matrixing approach. Of course, that also can be done, but it needs to be some justification why you need to do this. That can be done. Okay. This is what the very clear information about the matrixing approach. Correct? Now, there are some requirements when you can apply the matrixing, and there are some factors or design factors that where you can apply the matrixing, and very important is strength with identical or closely related formulation. So same requirement what we discussed during discussion of a bracketing, same discussion or same requirement is for matrixing. So strength with identical or closely related formulation means what? The formulation should be same, strength will be different, but the manufacturing from the same formula. That is what the first requirement. Second is what? Batches made by using the same process. So whatever the different batches are manufactured, they should be manufactured with the same process, same equipment, and should be packed in a similar container closure system, sizes or fields. Then only you will able to apply the matrixing approach, right? Hope everyone is clear with this. Very clear message, and my voice is also very clear. Words are very clear, right? Now here some design factors are clarified more. Okay, the next slide I would change, but it will take some couple of seconds. Okay, for matrixing at accelerated or intermediate storage conditions, means whenever you are doing any matrixing at the different conditions, care should be taken to ensure, please read this first paragraph very clearly. Testing occurs at minimum 3 time points. So when you have a stability study design, minimum 3 points should be tested completely, and in the 3 time points, initial and final should always be considered. So when you want to, suppose, let us example, 3 batches are there, right? And 3 batches, don't worry, I will be taking the example also, but I am just explaining you what does it mean. 3 batches are there, and 3 batches will have a 3, 6, 9, 12, 18, 24, 36. These many stations are there, batch number 1, 2, 3, right? So when you want to reduce the testing, then you need to ensure that initial means 0 and 36, all batches should be tested, and in between that you can have a testing at a 12-month, so that at least 3 time points should be tested completely. Then the remaining like 3, 6, 9, 24, these 4 station you can change or reduce the testing under the requirement for matrixing purpose. That is very clear. So I made it purposely in the bold letters. Okay, and this is not my requirement, this is the requirement of the guideline. When a matrix can design factors is applied, one strength or container size is no longer intended for marketing. Stability testing of that strength or container size can be continued to support the other strength of container sizes and or fill in the design. Very important, very good point. What happens sometimes, suppose there are 4 strengths, 1 milligram, 2 milligram, 3 milligram and 4 milligram. Now are all manufactured, but there is no intention to market the 4 milligram. There is only intention to market 1 milligram, 2 milligram and 3 milligram. So you are manufacturing 4, 1, 2, 3 and 4 milligram. Maybe in future you may intend to market 4 milligram, but today there is no intention to market this extreme of a largest strength. Take example, okay, I am giving example. Still, if you want to implement matrixing, you have to consider the 4 milligram for study, so that you will able to justify the intermediate levels of the complying to the requirements. So you cannot skip the highest strength, irrespective of whether it will be required for marketing or not. As you are applying the matrixing approach, you need to follow the this requirement, correct? So you have to perform the stability study of that strength, irrespective of whether it will be required for marketing or not, irrespective of that, right? Hope you are with me, correct? Very clear requirements are there. Now let us have some example of a matrixing designs. So when we are discussing about a matrixing design, there are two types of test reduction plan. There are two types of test reduction plan. One is, the first is a 1/2 reduction. First is what? 1/2 reduction plan. How you can follow the 1/2 reduction plan? Very simple example. I will again explain the same thing here. So there are three batches are there, right? Batch 1, 2, 3. And each batch is having there are two strengths S1 and S2. So there are two strengths S1, S2. Each batch is having a three different batches. Batch 1, batch 2, batch 3. And each batch is charged for the stability study for long term. Example is given for long term study. So there will be, what are the stations? These stations are in line with the ICHQ1D R2. If you need, how to be done and all, I will share the link. You can go through this. It is a short video about 10 to 15 minutes. So 0, 3, 6, 9, 12, 18, 24, 36. These many stations will be there at per ICHQ1D R2 for the particular batch. Hope you got the scenario. So there are two strengths S1 and S2, and each strength is having a three batches, and all are charged in the stability chamber for long term. Now you want to go for reduction, but with the half reduction, 1/2 reduction. So what is the requirement? Minimum 3 levels should be fully tested. So in that, initial and end of the shelf life is compulsory. So first of all, 0 means initial, 36 is the shelf life. All should be tested. After that, 1, which one is preferred? 12 month is preferred, because based on the 12 month stability shelf life determinations are there. So that is the reason most of the places you will find that the 12-month testing is followed for all the stations. So here in this case, 0 level all samples are tested, 12 month all samples are tested, and 36 month all samples are tested. Now you can see 3, 6, 9 and 18 and 24. These are the stations where we are reducing the testing. Now if you calculate here, why it is called 1/2 reduction, that I am explaining you. So if you calculate how much testing should be there normally without the reduction plan, you will come to know that 3, 6, 9, 18 and 24. So 5 stations are there, and total 6 batches. That means there are require how much? 30 testings are required, correct? But if you calculate here vacant places, you can see that 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. 15 testings are reduced. That is the reason out of 30 tests, 15 tests or 15 samples are reduced for testing. That is the reason this matrixing test reduction plan is called the 1/2 reduction plan. Hope you got the point from this simple example. You are with me, and very easy to understand. So this way also you can reduce the test plan. This test plan is called 1/2 reduction. The second test plan recommended by the guideline is a the second plan is a 1/3 reduction. 1/3 reduction. That was what? 1/2 reduction. Now this is the same example here. Whatever example we discuss in the 1/2 reduction, same example is displayed here. Only the testing is planned such a way that 1/3 reduction. So how? You can see here, same way, two strengths, each strength S1, S2, each strength is having a batch 1, 2, 3, and each strength is charged for stability study from 0 time point to 36 month time point. As per the requirement, same again it is followed. At 0, at 12 and at 36, you can see that all samples are tested. Now where we perform the reduction plan? We perform the reduction plan at 3, 6, 9, 18 and 24. These 5 stations we perform the reduction plan. So here if you count the blank positions, you will come to know that the 10 samples are skipped or 10 samples are reduced here. So total samples are supposed to test are 30, 30. Out of that, the uh, 10 samples we skip for testing. That is the reason it is called a 1/3 reduction. This plan is called a 1/3 reduction. But please understand this when you are reducing it. Please understand the logic here. Huh? See, you can see here batch 1 tested at 3 months. Batch 2 is tested at 3 months. Correct? So batch 3 is not tested here. Correct? But batch 3 of S2 strength is tested here. So that logic you need to apply. Same logic you have to apply when you discuss about the 1/2 reduction. Same logic is there. If you wish, I will again present the 1/2 reduction. You will come to know. Here the point is, all the different types of tests will be performed. See, at a 9-month, batch 1 is tested. You can see batch 3 is tested for S1. Then batch 2 is not tested, right, at 9 months, but batch 2 is tested at S2 strength. So that way you are not skipping any strength. That way you need to consider. So here the point is, when I am testing 9 months, I am skipping the batch 2 for 9 months. Then I have to consider the batch 2 of at a 9 month for strength 2. That way you need to consider. Similar logic is applied here in the 1/2 reduction. So you can just have a glance. If you want, you can pause the video and you can go through the this screen. So same logic is applied here. Just a minute, the slide there will be some couple of seconds. Yeah. So you can see here at a 3 month, you are testing this is a 1/2 reduction. Again, I went to the previous slide. So at 3 months, I am testing the batch 1 and batch 2 of a strength S1. I am skipping the test for batch 3. Correct? So then I have to again go some other place where I will perform the testing. Hope you got my point. So same way, same logic you need to apply, that all the testings are covered. Okay. Now the other matrixing approach is the time points plan. The other matrixing approach is what? Time points plan. But again, I am telling you that whatever I am discussing, the time points plans and reduction plans and all, then you need to have a justification for that. Okay. And that should be specified in the stability study protocol. And when you are submitting the stability studies data to the regulatory agency, you have to specify that whether you have followed bracketing or matrixing, at what extent you followed bracketing and matrixing, what is the rationale for that you have to submit to the regulatory agencies along with the submission of a stability study protocol and stability study data with the regulatory agencies. Hope you got my point. So now this is a time point plan. So here what happened, you can see here now here two scenarios are considered here. So there are 3 strengths, S1, S2, S3 strength, and each strength is having a different pack style, A, B, C. And let us consider that A, B and C packs are different packs, and each pack is having a separate batch number, batch A, 1, 2 and 3. So here this time points plan, here all combination of batch, strength and container size are tested under this plan. What is considered here? All combination of strength, batch and container size are tested. How? You can just consider here batch number 1, sorry, T1 is what? Batch number 1 is tested for container size A. Correct? You can see here batch number 2 and batch number 1, batch number 2, batch number 3, T1, T2, T3 are here considered. Now if you consider T1, you can see here the container B for batch number 3 is tested. Container A, batch number 1 is tested, followed by batch number 3, container B is tested. Means batch number A is also container A is also respected, container B is also respected, and similar way batch 2 you can see T1 only. Yeah? You just track one T1, or you track T2, or you track T3, whatever, and you just track how it is it is combined for batches and strength and containers. You will come to know. So if you track any T1, T2, T3, any one of three, you will come to know. Like T1, T1 is what? Batch 1 container size A is tested. Then batch 3 container B is tested. Then batch 2 container C is tested. That means what? All container size, all different strengths, and all different pack or all different batches are considered under this scenario of a matrixing approach. This also can be justified and implemented by many companies. But the next whatever I am going to present, it is somewhat risky, you can say, to implement and to follow. This is my personal opinion. However, this is recommended in the guideline. But the point here is, you can see here, when we are considering the time points plan. So here we are reducing the time points also when we are discussing about the same. It is the same scenario what we discussed earlier. Same scenario. Hope you are not getting confused. If you are getting confused, pause for some time, put your thought process, you will understand. Okay, what I'm telling here, same scenario, three strengths, three pack style A, B, C, and each pack style is having a batch A 1, 2, 3. So here what happened? Here only certain combination of batch strength and containers are tested, and certain combination of batch strength and container are not tested. So when this we are following, this matrixing approach we are following, so here some risks are there to justify. It requires a lot of justifications, because we are completely skipping here, you know. Like T1, here you can see batch 1 container size A is tested, right? Then here you can see the container size B is not tested at all. So you are completely skipping the test of container size B. But yes, container size C is tested for batch 2. So that way, there is a skip testing. You can see further time points also, there are some skip points are there. So basically this matrixing, whatever I am presenting the example for time point plan and factor with respect to the strengths and all. So here we are, skipping some strengths for testing purpose. So when you are preparing this plan, then you need to be very clear with respect to the statistical approach, that which test I am skipping. If I am skipping that particular time point, or if I am skipping particular batch, or if I am skipping particular pack, or I am skipping particular strength, then how it will be justified by considering the other batch or other pack or other strength results. That you need to be have a thought process. So basically, this type of matrixing you can very well follow when you have a already develop a product. You can say the product is running since long in the market, you have a sufficient stability data and all. Then definitely you can reduce this type of matrixing approaches in your reducing the stability approaches by following this matrixing approach in future. But when you are considering the submission purpose, when you are initially submitting the data for regulatory submission, then personally, what if you ask me personally, I don't recommend. But yes, you can follow unless and until you have a sufficient data, sufficient variability parameters and all. You can follow this parameter also, there is no any problem. So that is what the next point we are going to discuss here. And the next point is degree of reduction and its applicability, because this is what the point we need to understand, that when we can apply and how, at what extent we can reduce the testing. You got my point? See, I am trying to put here in the pointer, but the point is you may not able to see the pointer in the, I don't know, but that is the reason I am making use the word of clear cut words I am reading, all the words, so you please focus, right? I am not able to do here on my screen. So what is the applicability and degree of reduction, right? So we need to consider when we these points when we are considering the matrixing. So what we are considering, knowledge of data variability, very important. Knowledge of data variability. Now all the inspectors are focusing on data variabilities and all, and that is the now current focus of all the regulatory agencies, including USFDA, EU, WHO, and all. And that is the reason I have planned the paid training session on a continued process verification. That training is focusing on that expectations of the different regulatory agencies and about the variabilities and all. Okay, because this is the current expectations of the people. So the point is knowledge of data variability. Second is expected stability of the product, means what is the expectation of that stability of the product, that you need to know based on the development data. Availability of the supporting data, so how much data you have supporting data, and what is the possibility of a variation impacting on the stability study, that all data you need to have. Stability differences in the product within a factor or among factors, so that is what I was telling you. So when we are performing the stability study, so there are different factors, storage conditions, period, and all. So what is its impact, that we need to aware about this. Number of factor combinations in the studies. So when we are performing the study, what are the different factors and what is their combinations and what are their expectations, expected results, that we should able to know. So basically, when you have a maximum data, or you can say the maximum stability data, then definitely this is very easy to implement. Matrixing approach is very easy to implement. A matrixing design is applicable if supporting data indicate predictable product stability. So supporting data indicates predictable product stability. How, what is predictable? Means, like suppose I'm having the stability study since last so many years, and I know that the example I'm telling you, that assay at initial at suppose 100%, then it will be slightly reduction in the assay, 99.8, 99.7, then 99.5, then 99.0, then 98.5, and up to the end of the shelf life it will be suppose 97.5 or 8, like that. Example I'm telling you. So it is a predictable. And same way, if the assay is reducing, there may be increase in the impurity. That impurity also may be the known impurity, and it is well within the acceptable limit. So this is what the predictable, that assay is slightly reducing up to the end of the shelf life, and with the same exponentially the impurity is also increasing. So when I will come to know this predictable, this is called the predictable stability study. When I will come to know that this particular product is having a predictable stability study, then definitely we can follow the matrixing approach. Matrixing is appropriate when supporting data exhibit only a small variability. So very small variability is there. When you come to know, then definitely you can follow the matrixing approach. Correct? Hope you are with me and you are understanding the points, right? Further, however, where the supporting data exhibit moderate variability, matrixing design should be statistically justified. But when you are evaluating the data and you come to know that there is a more variability, moderate variability, means somewhat more variability it has somewhat more level, then you need to evaluate the matrixing design based on some statistical approach. So matrixing design should be statistically justified, and then only you should follow the matrixing design. But if there is a very little variability, then definitely you can follow the matrixing approach. Okay. Any reduced design should have the ability to adequately predict the product shelf life. So this is what the same thing is there. So whatever you reduce the shelf life, you should able to predict. Whatever you reduce the testing, sorry, whatever you reduce the testing, you should able to predict the shelf life. Suppose you are reducing, suppose 3 months test, but the next batch you are testing at a 3 month. You skip the test of 3 month for batch 1, but you performing the test of a batch 2 at a 3 month. So that way, you should able to predict the stability steady shelf life, so that if batch number 2 is having a proper shelf life, automatically batch 1 will also have. And finally, anyway, you remember the matrixing approach. At the end of the shelf life, we need to test all the samples. So that way, again indirectly you are performing the samples testing at the end of the shelf life. So that again added the advantage to support the shelf life determination. So that way, you can apply the reduction techniques for testing in the reduction for the stability studies, right? But there are some risk. Now when you go through this guideline, when you go through the guideline, the point here is the heading you will come to know that risk with respect to the matrixing. There is no risk with respect to the bracketing basically, because bracketing is very easy, very, and can be implemented. No much justifications are required, rationales are required. But because when you are submitting the data, along with that you are submitting the manufacturing formula and everything you are submitting, packaging profiles and all. So automatically this is understood by the reviewer also. But when you are reducing the testing at matrixing, then there are some potential risks. So what are the potential risks? They are listed here. Less precision in shelf life estimates and yields a shorter shelf life. Of course, there is a less precision because there are a lot of parameters. First of all, variabilities, estimates and all. Second thing, statistical evaluations. Third thing, if that also you perform, and if suppose there are some adverse trends and all, then it may impact on the assigning the shelf life. So assigning the shelf life will not be directly in line with the ICHQ1E, but then you need to follow the statistical approach, and which can lead to the shorter shelf life. That is the main risk with respect to the matrixing. So that is the reason many people performing the matrixing at the commercial stages, not at the submission stages to the regulatory agencies. But again, it depends on the availability of the data. Okay, I am not giving a rule, but most of the practices that I'm sharing you. Insufficient power to detect certain main or interaction effects, thus leading to incorrect pulling of data. So what happened, as there are little data, because we are reducing the test, so data will be limited. So there will be insufficient power to detect the data, where there will be some adverse trends or where there will be some failures. So due to that, it will be creating the problem during the shelf life estimation. And third, if there is an excessive reduction in the number of factor combinations, if you follow the excessive reduction, so whatever I have given, I have displayed the examples for the matrixing and all. So these are you can say these are some the examples. You can follow more reduction techniques also based on your product and all. But the point is, if you follow the excessive reduction in the number of factor combinations tested, data from the tested factor combination cannot be pooled to establish a single shelf life. It may be impossible to estimate the shelf lives for the missing factor combinations. So if you miss any factor combination under these, you may not able to assign a proper shelf life. This is actually the problem when you perform a maximum reduction in the sample testing. What happened when we start putting a thought process for reducing the testing and all, then further the people will ask, then why you are performing this test, why can't skip that also, why can't skip that also. And that in that case, you may not able to justify and you may skip that test. But the problem is, if you follow the excessive reduction in the test, then you may face a lot of problem. So when you are following you are excessive reducing the test, try to follow some statistical approaches to design your strategies, right? This is what point what I want to share with you, correct? So about this is what I was telling you, that for variabilities and all, I already told you that, this topic is very, very important with respect to the continued process verification. And I am going to discuss this complete webinar for 2 days on continued process verification on these days. You know, uh, you can go through the things if you are interested in the paid trainings. You if you are interested in your company bookings also, you can arrange the training to your all uh, big team so that you can reduce the cost. Because whatever I am conducting this paid training, basically I am collecting the fund to support the farmers. So this is a 2 days full day training program where you will get a learning. I will share the training material, guidelines and all. You will get the certifications, and whatever the money you will be paying as a fees, that I will be collecting and I will be donating to the farmer. So you will be indirectly supporting for the noble cause. So before this, we already successfully completed the one training session to support the farmers. That was the quality risk management. And this training session is on a continued process verification. There are a lot of myths, even many companies not implemented properly. So how you can practically implement at your end, and what are the practical problems and how we can resolve that, all we were going to discuss during this session. If you are interested in more about this topic, you please write to me. I will share the details, agenda, and what we are going to discuss, that I will share with you. The next thing I just want to share is the YouTube membership program. So under this YouTube membership program, there are three categories: GMP, CGMP, and advanced GMP. So if you are the member, then you will get a very good discount on these training sessions. And second thing, there will be a lot of benefits when you click under GMP. There will be some benefits are given. If you click CGMP under the join button, you will get some different benefits, and same way for advanced GMP. But what I suggest that the membership is a paid, but the subscription is totally free of cost. So you can subscribe this YouTube channel, so that you will be subscribing the free learning platform where you will get all the free learnings from the different topics, right? So many people not yet subscribe this YouTube channel, so I kindly request all of you to subscribe this YouTube channel. Also please share this link to all of your colleagues, so that they will also get learning from the uh, this particular video, right? So now uh, this is what I just want to share with you. And thank you so much for your kind attendance. These are my contact details. If you go to YouTube, you just put my name, Hitendra Kumar Shah, you will get lot of learning videos. Now let us discuss about some questions, and I will share the training material. Just a minute. So the question is, Mr. Bijay Ghosh is asking, in case of API vendor change, is one batch instability is enough or not? See, basically, when you are changing the API vendor, so you need to evaluate that what exactly is changed. Only the vendor is changed, whether the manufacturing process, impurity profiles, or everything is same, or any CP nature is same, that all you need to evaluate. Based on that, you need to take a decision, okay? But if there is some mode of synthesis is different and all, then I don't suggest to have only one batch. You need to have more batches. So that is what I just want to share here. Mirin Patil is asking, sir, if only one or two excipients change, then see, if in case of a formulation there is a slightly change in one or two excipients, maybe suppose the concentration or ratio is changed, then definitely you can go ahead. And if the excipient itself is changed, but if the excipient is of some category of the excipient is changing, or complete change of the excipient, still you can go for the approach, no problem. Only you need to provide the justification that how it will behave the same. For example, suppose there are the three strengths, 1, 2 and 3 strengths, and if in the third strength there is some different excipient is used, so if I am performing the testing at a strength 1, how it will behave the similar for strength 3, irrespective of that particular excipient, that you need to justify based on the development data. And definitely you can follow the bracketing and matrixing approach. Mr. Milind Patel is asking, kindly please send the link for the PPT. Yeah, I am sharing the link. Don't worry. So please be with me. But my request is, when this link, you can download the training material from the link. But my request is, please download the training material immediately, because the link will expire in just a couple of days. Okay. The second thing is, what I just suggest that, when you want to know and learn, please go through the recorded webinar. The reason is, training content is very small, but lot of points are discussed which may not be part of the training presentation. So that is the reason I always suggest to refer the recorded webinar. But yes, for your reference, I have shared the link. You please download it so that you will have the training material. And after that, there is no any question. Mr. Shishir Kumar was asking, please use pointer. But sorry, my dear friend, what I was discussing during the discussion, that I am not able to use the pointer because I need to use some interface between the YouTube and my presentation. So I am putting my pointer at interface, which is not visible in the YouTube. That is what the problem. But don't worry, if you have any questions after this also, put your questions in the comment section. I will reply to all your questions. Thank you. Thank you so much. I think there is no any questions. So thank you so much for your kind attendance, and hope you subscribe the YouTube channel. If not yet subscribed, please subscribe. And I kindly request you to share and to inform to your team members to please subscribe this YouTube channel, so that everyone will get the benefit from this learning. Thank you. Thank you so much, and ensure all time compliance. Thank you. And now I am putting off the streaming. Thank you so much.